Hey everyone, we're solving this bio paper today, biology after a long time. This is May-June 2022, 9700 paper 1-1. So from 2020 onwards, you're gonna have 1 hour 15 minutes instead of 1 hour. And I guess the exam for the October November session is on the 16th. Okay, so I'll try to solve all 3 papers by then. Okay, and if you do have other requests, please feel free to comment below. And remember to subscribe to the channel if you like the content. So let's begin. Before we start, I just want to take a look at the threshold for this paper. So, one sec. Yeah. So the maximum mark is 40. My bad. Excuse me. So, A, B, C, D, E. So for paper 1-1, what do we have here? A was at 30, B was at 25. A was at 30, B was at 25, C was at 21. D was at 18 and E was at 15. D was 18 and E was at 15. My bad. So basically, if you look at the difficulty of all the papers of all these three, it seemed like paper 1-2 was the hardest. Paper 1-2 was the hardest. Since A was at 27, but paper 1-1 and 1-3 were on the easier side, right? So let's begin. Let's see how we deal with it. Let's begin. A student used a stage micrometer scale to calibrate an eyepiece reticle. The diagram shows the view of both the stage micrometer scale and the eyepiece reticle seen by the student. The divisions on the stage micrometer scale are 0.1 millimeter apart. The student removed the stage micrometer scale and viewed a slide with blood cells on it. The same lenses were used so that the magnification remained unchanged. The student measured the diameter of one of the white blood cells on the slide using the eyepiece reticle and recorded that it was 8 eyepiece units. What is the correct diameter of the white blood cell? So basically, guys, we use the stage micrometer for calibration with the eyepiece reticle. Because the stage micrometer is usually larger, the divisions, the graduations are larger. That is why we can't use the stage micrometer directly to find out actual lengths. Okay, so basically we know that the divisions on the stage micrometer are 0.1 millimeters apart. So it basically is equal to the length between 50 and 90. So 0.1 millimeter is the same as this length from 50 to 90. So 50 to 90, 90 minus 50 is basically 40 EPG units. So 40 EPG eyepiece reticle units is 0.1 millimeter. So 1 EPG unit will be 0.1 by 40 millimeter. So the WBC, which has 8 EPG units, will be 0.1 by 40 times 8 millimeter. That is 0.1 by 40 times 8, which is 0.02 millimeter. Now you guys know how to convert millimeter to micrometer, right? It's basically into 10 to the power 3, which is 20 micrometers. So the answer should be 1C. All right, so please check. Yeah, the answer to 1 is C. Let's move on to number 2. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed below. mRNA passes through the ribosomes, produces through to the ribosome. It's basically the pore in the nuclear envelope. Produces the mitotic spindle during cell division. Packaging of hydrolytic enzymes that will remain in cell. So 3 is Golgi body basically. 3 is Golgi body, or the Golgi apparatus, and this is the pore in the nuclear envelope and produces the mitotic spindle. So this is basically the centriole. Okay, this is the centriole. The centrioles and the centrosome is the location of the centrioles. Okay, there are two. So the appearances were listed by letter. So let's see. So the centriole is a non-membranous organelle. Okay, so 2 has to be non-membranous. So it has to be W or Y since the centriole is non-membranous. Okay, so that doesn't help. Now the central is actually cylindrical. It is not spherical. That is why I'm going to exclude A and C. It can't be W. It has to be Y. Next, it's between B and D then. So the Golgi body. It's basically a membrane-bound sac which is arranged in flattened stacks. So 3 should actually be Z. That is why I'm going to opt for D. Also, you can check X. X is a double membrane interspersed with pores. Yeah, that's the nuclear membrane, the nuclear envelope. Okay, which size range would include most prokaryotic cells? Okay, prokaryotic cells are typically prokaryotic cells are typical. Like, what about a eukaryotic cell? It's usually around 40 micrometers usually, and prokaryotic cell is usually around 40 to 100, and a prokaryotic cell is usually around 1 micrometer. So the answer should be B. Okay, it isn't in the nanometer range. All right, so the answer is B. You can check more of this in the course book. Basically, see this in chapter 1. You'll have the size given. Here you go. So they're usually around 1 micrometer, 1 to 5, and the eukaryotic cell is usually around 40. All right, that's what you need to know. 4. What is present in a typical prokaryotic cell and typical eukaryotic cell? Okay. No, both of them do not have 70s ribosomes. Eukaryotes have 80s. But here's the tricky part. Basically, in eukaryotic cells, we also have mitochondria and chloroplasts. And according to the endosymbiont theory, they were prokaryotes at one time. So this was a trick question. Eukaryotic cells also have 70s ribosomes. Even though mainly we have 80s, but 70s ribosomes are present in the chloroplast and mitochondria. Not all eukaryotic cells have starch. We do not have circular DNA in the cytoplasm. Prokaryotes do, and yeah, prokaryotes do not have. Centrioles are not present in all organisms. Understood? Like centrioles are missing in plants. Special for animals. Which statement of viruses is correct? They all have a capsid made up of a protein. They do. That is a part of the virus. It is true. Some virus may have DNA or RNA. They all have an outer membrane of phospholipase. This is wrong. They all contain ATS ribosomes. Wrong. Samples of glucose, sucrose, and a mixture of glucose and sucrose were divided into two halves, M and N. AM was then tested with Benedict solution. N was boiled with dilute hydrochloric acid, neutralized, and tested with Benedict solution. Okay, so that's the test for reducing sugar. Non-reducing sugar, I mean. We break it down and then it becomes a reducing sugar. The color of the solution was compared to color standards. Which table identifies the correct color change for these samples? So glucose, sucrose, and a mixture of glucose and sucrose. Okay. So M, if we test M with a Benedict solution, so we're gonna get a positive test, but it will only be due to glucose. Okay, it will only be due to glucose and the mixture, slightly due to the mixture. Okay, so basically I'm going to tell you something. A is wrong because this part can't be blue. This part can be blue. It has to be like, the color change, right? It's going to go from blue to green to yellow to orange to a brick red precipitate. So this can be blue because we do have glucose here, so we would get a positive test. Similarly, B is also wrong because, okay, this is yellow, but this should also be colored, because in the mixture we do have a reducing sugar glucose. That's why these two are wrong. Okay. Now what about C and D? This looks good, guys. This gives us a yellow color. This is also yellow. This is yellow. This is yellow. Great. In sucrose, we're gonna get a blue color. This is a must because it's a non-reducing sugar. Now in N, basically what happened was we've boiled it with diet HCl, so we've actually hydrolyzed the sucrose. So now everything is that sucrose is made up of glucose and fructose. So all of them are reducing sugars right now. That's why afterwards, what's gonna happen is in the boiled solution for N, since we have both glucose, then a lot of glucose due to the breakdown of sucrose, and we have fructose, all of them are reducing sugars, we should be getting a red color. We should be getting a red color. Understood? Because we have a lot of reducing sugar. Now let's see between C and D, what's the better answer? Here in the mixture we get red. This is correct because we have a mixture of everything. That means that there's a high concentration of reducing sugar. Now between these two, our decision comes down to this. Basically, the sucrose, when it breaks down, um, uh, basically from blue to red is an exponential change. I don't think we have enough reducing sugar to produce this change. Like from blue, it might go to yellow, but going to red is a bit too much. So the answer should be C. This is the best answer here. And glucose, glucose will remain yellow. Like even if you hydrolyze it, right? Like glucose can't be hydrolyzed. It can't give a better color change. It will still remain yellow. So do you guys understand the difference? Because this is actually the determining factor. Glucose can be hydrolyzed. So even if you do hydrolyze it, or if you don't, the color change will remain the same. So this can't be possible, right? So this is a determining factor. But if you break down enough sucrose, it might have turned red. So actually this is not the determining factor. Rather than that, the first one, glucose won't turn red. All right. Number 7. Wait, what? The options haven't loaded here actually. Weird. Is everyone facing this problem? What even? Wait up. Let me check the main paper. The 976. Yeah. Weird. Yeah, in the main paper it hasn't. It hasn't been printed properly. That's weird. Well, let me check another site. Extremely weird. This is QP11. Yeah, it's the same for every paper apparently. Right, so no worries. Basically, which molecule contains 1,4 glycostatic bonds? Oh, the options are here themselves, I guess. So 1,4 glycostatic. Cellulose actually contains beta 1,4 glycostatic bonds. Okay. Amylose contains alpha 1,4 glycostatic bonds only. It doesn't contain alpha 1,6. That's amylopectin. It contains both. Glycogen contains both 1,4 and 1,6 alpha glycostatic bonds. So all three of them do contain 1,4 glycostatic bonds. It wasn't mentioned whether it was alpha or beta, so C is the answer. 8. The diagram shows 3 triglycerides, X, Y, and Z. Which one is correct for these triglycerides? Contains saturated fatty acids. Okay. So basically, these two are unsaturated. mainly. Okay, but look at this side chain over here. This one is saturated, this one is unsaturated, this is one is unsaturated, unsaturated, saturated, unsaturated, saturated, saturated, saturated. If you have the presence of a single double bond, that means it's an unsaturated fatty acid. So actually this was a trick question. All three of them do contain at least one saturated fatty acid. However, only X and Y contain unsaturated fatty acids. Z does not contain an unsaturated fatty acid. Okay, that is why the answer should logically be A. Contain more than two different types. So that's X and Y, okay? Hopefully that makes sense. 9. Some foods contain hydrogenated vegetable fats. These are unsaturated fats that have been converted to saturated fats. Which property of the fats will have changed? So basically, if you convert an unsaturated fatty acid to a saturated one, do you see these kinks on the chain? These will become more straight. As a result, they will fit closer together, but beforehand the fit wasn't that great because it was bent, there was a kink. So honestly, after conversion to saturated fatty acids, the hydrocarbon chains fit more closely together. Okay, but the solubility in water actually decreases. They will have less double bonds and they will remain liquid. No, unsaturated fatty acids have a tendency to remain liquid, but saturated typically remains as a fat. A polypeptide chain contains a specific number of amino acids, and how many peptide bonds are present in this polypeptide? Okay, so check this, guys. Suppose if you have two amino acids, you're gonna have one bond. If you have three amino acids, you're gonna have two bonds. So do you guys see the trend? So depending on the number of amino acids, if you have two amino acids, one bond. Three amino acids, two bonds. So it's basically the formula is n minus 1. The number of bonds will be 1 less than the number of amino acids, okay? So the number of amino acids will be 1 more than the number of bonds. Which statement is correct? Amylase, ribose, and phospholipid are all macromolecules. Cellulose, glucose, and catalase are all polymers. Deoxyribose, fructose, and ribose are moles. Okay, let me cut these according to the options. So, my bad. Basically, guys, sucrose is a disaccharide, so D doesn't make sense. Amylopectin is a polysaccharide. Deoxyribose isn't one either. Cellulose is a polymer. Glucose is not a polymer. It's a monomer. Amylase is a protein enzyme. It's a macromolecule. But ribose is just a sugar. So it's not the answer. So the answer has to be C. Deoxyribose, fructose, and ribose are all monosaccharides. Correct. 12. A student used colorimetry to monitor the hydrolysis of protein by a protease enzyme. The student used biuret solution to determine the concentration of protein in the hydrolysis reaction. The student produced a calibration curve using known concentrations of protein. Which diagram shows a calibration curve? So guys, in a colorimeter, we actually can measure transmission or absorbance. Check this. For the biuret test, it's the test for proteins. Okay, so you guys need to know about the biuret test properly. Where is it in the course book? Um, Presence of starch, presence of lipids, here you go. So in the biuret test, the nitrogen forms a purple complex with copper 2 ions, and this forms the basis of the biuret test. So the reagent is the biuret reagent, and you can use it as two separate solutions. It's basically potassium hydroxide or sodium hydroxide and copper 2 sulfate, which is blue in color. Alternatively, there is ready-made biuret. So we mix them, and to stop the copper ions reacting with the hydroxide ions and forming a precipitate, this ready mixed reagent also contains sodium potassium tartrate or sodium citrate, okay, or else a precipitate will be formed of copper hydroxide. You know that from chemistry, a pale blue PPT. Now the biuret reagent is added to the solution to be tested. No heating is required. It occurs at room temperature. A purple color indicates that the protein is present. The color develops slowly over several minutes. Alright, so the greater the protein concentration, the greater the protein concentration, the greater the purple color. Okay, the greater the intensity of the purple color. So if the test tube is very purple, basically if you pass light through it, all of it will be absorbed. Absorbance will be greater. So light cannot really pass through the other end. So the greater the intensity of the purple color, the greater the absorbance. Absorbance means the tendency of a solution to absorb light. So it doesn't basically allow light to pass through it. So transmittance would be 0. So the graph would actually be like this for transmission. Okay, it's actually the other way around. Anyway, a student completed an experiment to measure how increasing concentrations of substrate affects the rate of an enzyme controlled reaction. The student then repeated the experiment after adding a fixed quantity of a reversible competitive inhibitor. Which row describes the effects of a reversible competitive inhibitor on the enzyme activity? So basically, reversible competitive inhibition can be reversed by increasing the concentration of the substrate. So if you increase the substrate concentration, the rate will increase again. Okay, so 13 is between B and C. In non-competitive, there will be no effect, little effect. Now, where does the inhibitor bind? It actually binds to the active site itself, not the allosteric site. Okay, that's why it can be reversed. Now 14, it shows a liposome. Liposomes can be used to move therapeutic drugs into cells of the body to treat conditions such as cancer. You will learn more about this in A2, in genetic engineering. So we use it for therapeutic uses. It has therapeutic uses. So basically, we transport the drug here, which is actually, like it allows transport because the outer region, the heads are hydrophilic in nature. So even though the drug, you know, might be susceptible to water or it may be hydrolyzed, it can be prevented by keeping it inside. We keep the drug inside to prevent hydrolysis. Okay, you guys need to understand that. But there's this thing that, like, it depends. You can even keep the drug here or here. So you guys know that these heads are hydrophilic in nature. So if you transport the drug in location 2, the drug's nature will actually be hydrophilic. This is hydrophilic. And the drug located here, the tails are hydrophobic, right? So in 1, the drug has to be hydrophobic. So it's hydrophobic in location 1 and hydrophilic in location 2. So the answer should be C. Some processes occurring in cells are listed, which uses ATP. This uses ATP, this uses ATP. Facilitated diffusion is a passive process. Phagocytosis requires ATP. So it's actually 1, 2, and 4. 15 should be B. Okay. Diffusion, normal diffusion and osmosis are passive. Active transport and all bulk transports are actually require energy. The graph shows the change in concentration of a solute inside a cell. What explains this change in concentration? Okay, so basically, yeah, something can diffuse out of a cell. If you actually if endocytosis occurs, the concentration would actually increase inside the cell. So 2 can't be the answer. I'm going to exclude 2. Is between B and C. So 3, exocytosis, true. Exocytosis gets rid of something from inside the cell. So this is right. Osmosis may also be correct. Okay, 1 and 4 are true for both things, like either into the cell or outside the cell. But here the main factor was endocytosis has to be false because we are measuring the concentration inside the cell. Endocytosis will actually increase the concentration inside the cell, but here we are actually decreasing it. So our answer is in fact 16 is B because 2 is wrong only. Next, 17. The indicator cresol red changes from red to yellow when put into acid. 4 blocks of agar containing cresol red were cut to different sizes, measured in millimeters. The blocks were submerged in acid. All other variables were kept constant. The time taken for each of the blocks to completely turn yellow was recorded. Which of the 4 blocks became completely yellow most quickly? Okay, so this depends on diffusion, and diffusion depends on the surface area to volume ratio. Okay, so let's check. So 3 into 30 into 30, 3 into 30 into 30, that's 2700. Now the formula for surface area is base area 2 times base area, or basically we can't use this since we do not have the okay, 2 times base area plus perimeter into height, you could do that. Or basically just you could add up 6 base areas. It's basically 2 times, are these the dimensions? Yeah, these are dimensions measured in millimeter. Okay, let's do that. 2 times base area plus perimeter of base into height. So this is the block basically. So what if the base area is this 30 times 30? Okay, so that's basically 30 times 30 into 2, which is 1800, plus perimeter of base 30 into 4, which is 120, times the height, which is just 3. So that's 1800, 120 into 3 plus 1800, that's 2160. So the surface area to volume ratio is 2160 by 2700, which is 0.8. The ratio is 0.8. So that's it for A. Let's look at the other ones. So here the volume is actually 6 into 6 into 6, which is 216, and the okay, so here we could find out the area pretty easily. 6 times 6, that gives us one area, that's 36 millimeter square, times 6 um, times 6 gives you the area. So 36 is the area of one side. Okay, so if you multiply this by 6, you're gonna get the surface area, which is 1 is to 1. It's basically 1 is to 1, the surface area ratio to surface area to volume ratio. All right, but you get this question quite often. Honestly, rather than measuring the surface area to volume ratio in D, basically in D you're gonna get the same surface area to volume ratio as B. It's going to be 1 because the volume is 12 into 12 into 12, 1728, 288. Well, let me try this. Yeah, this is also 1 is to 1. What about C? This is 864 the volume, and the area is 12 into 12 into 2, 288, and 12 into 4 to 6 plus 288 is 576. This is actually a 0.67 is to 1. So yeah, there are some confusing questions about this type of question 17. They were submerged in acid. Now all other variables were kept constant. So for these questions, typically the trick is the one with the shortest dimension. You won't look at the surface area to volume ratio. Rather than that, you just have to look for the small one with the smallest dimension. I've done questions like these in 2021, I think, the year 2021, where I told you that the one with the shortest dimension, at least one, will be the winner. It will, diffuse through it the fastest. The surface area to volume ratio is important for other things. When the lengths are proportional, really. But just think about it. If you have one end which is very small, it's very easy to penetrate through that area. Do you guys understand? So that's why 17 is in fact A. Although the highest surface area to volume ratio does go to B or D. Okay. Which process requires mitosis? 18. One sec. Repair of cell structures, reproduction of unicellular eukaryote, the growth of multicellular organisms from a single cell. Cloning requires mitosis. Repair of cell structures by protein synthesis. This can occur naturally without mitosis, so 2 is wrong. Growth of multicellular organisms from a single cell, true. Like from we became embryos from zygotes. Reproduction of unicellular eukaryotes. That is also mitosis. So it's 1, 3, and 4. 18 is B. Wait. 19. Which events listed are part of the cell cycle? All three. We know the cell cycle mainly has two parts. We have interphase over here, and then we have mitosis over here, and we have cytokinesis. So interphase has the G1 phase, the S phase, G2 phase. There is G0 in some cells. We have mitosis, which consists of prophase, metaphase, anaphase, and telophase, and then we have cytokinesis. Okay, so all three are correct. 20. Telomerase is an enzyme that adds nucleotides to telomeres. Which statement about telomerase is correct? Telomerase actually prevents the breakdown of the telomeres, prevents aging. A high concentration of telomerase in cell damages DNA during DNA replication. This is rather the complete opposite. A high concentration of telomerase in cancer cells limits the rate of tumor growth. The low concentration of telomerase in stem cells means that these cells can divide an unlimited number of times. This is wrong. If it was high, it would have been true. The low concentration of telomerase in body cells mean that these cells can divide a limited number of times. This is true. Also, number B, a high concentration of telomerase in a cancer cell limits the rate. No, it actually facilitates the rate of tumor growth. Okay, so high telomerase means more growth, less telomerase, low telomerase means less growth. 21. The photomicrograph shows cells at different stages of mitosis. Okay, guys, so what is happening over here? Cell P shows anaphase. Clearly, it's anaphase. It's not telophase though, it's moving towards it. T is basically the T is metaphase, right? T is metaphase. P is anaphase. R is prophase. Q looks like telophase or cytokinesis, something like that. So the correct order should be, oh, and S is just, it's preparing. Okay, so what's the actual um, process? S is just before prophase. It's in interphase right now. So what's the correct order? Interphase followed by prophase, which is R, followed by metaphase, which is T, followed by P, which is anaphase, and then we have whoa, wait up, and then we have telophase Q. So SRTPQ, SRTPQ. This is correct. 4 is correct. I'm gonna go with 4. A is wrong. Cell P shows anaphase, which is also true. 1 is also true, so this has to be wrong. It's between B and C. Spindle formation is occurring in cell Q. What? Cell Q is, it's impossible. Because where does spindle formation occur? Telophase is like the end stage, right? So spindle formation actually begins occurring in late prophase and early metaphase. Late prophase basically. So it should have been R, which is why 2 is wrong. I'm not going to take 2, which is why the answer is C. The amount of DNA in cell R is the same as in cell T. R is the same as in cell T. True, true. They both have double, right? See, they have double the DNA. If it was S, maybe it would have been wrong, because mitosis has already taken place. 22. Bacterial cells with DNA containing only the heavy isotope of nitrogen are allowed to reproduce for 3 generations. In a culture medium containing the normal isotope nitrogen 14. Okay, so guys, I have a diagram for this actually. You guys should remember this. Check this. Here you go. Whoa, wait up. So yeah, basically in the first gen we have 100% nitrogen 15. In the second gen, you know that DNA replication is semi-conservative, right? So one strand will be new, one strand will be normal. So in the first gen, what's the first gen actually? What's the first gen? You guys need to understand that. What is the first gen? Vector cells with DNA contain only the heavy isotope of nitrogen 15 and are allowed to reproduce for 3 generations. So even though this is the first cycle, after the first cycle, we are going to get the first generation. This is the second generation, and this is the third generation. Okay. So in the first generation, we have 50% nitrogen 15 and 50% nitrogen 14. However, both the DNA molecules contain the so what's the question? Which percentage of DNA molecules produced? DNA molecules means 2 strands, okay? Which percentage of DNA molecules produced contains strands with the heavy isotope? See, both strands contain the pink strand. Both DNA molecules contain the pink strand. So 100% of the DNA molecules contain the pink strand or contain nitrogen 15. Following that, this will divide into 2 stages. Now, look at this. The pink strand only exists in 2 of the 4 ones in the second generation. That means 2 out of 4 means that's basically 50% of the DNA molecules. So it should be 50%. And at the end stage, look, only 2 out of 1, 2, 3, 4, 5, 6, 7, 8. 2 out of 8, which is 25% hybrid DNA. So here we have 100% hybrid DNA. Here we have 50% hybrid DNA. Here we have 25% hybrid DNA. That's what this means. They actually meant hybrid DNA. This is a common question, okay? You'll find other variations soon. A bacterial circular DNA molecule is 26000150 base pairs long. 26% is adenine. So what about cytosine? So we know that adenine binds with thymine, so it's 80. So 6% is adenine. Thymine will also be 26%. So 100 minus 26 minus 26 gives us 48%. So 48% will be divided between cytosine and guanine. 48 divided by 2 is 24. So Cg, that's 24, 24. How many cytosine bases would be in the DNA molecule? Okay. So that's 24 by 100%. 24% into 2600150. That's basically how much? So you might get tricked here. If you just multiply 24% into 2600150, you're gonna get 624036. Basically, like, check this. This is the tricky part. We have a bacterial circular DNA molecule which is 2,600,150 base pairs long. Okay. So here they're talking about base pairs. Okay. So how many? So if you have 2600150 base pairs, that actually means you have 2600150 into 2 bases. So that's what the question asked. How many bases do we have? So since 26% of the bases are adenine, 26% are also thymine. That means C and G are 48%. So C is 48 by 2, 24% of the total bases. So if you multiply this by 24%, you're gonna get your final answer. 24 by 100 into 2600150 into 2. So you're gonna get a final answer of 124807. Tricky. Most candidates would typically choose A here. Okay, be careful. 23C. Which statement relating to the structure of DNA is correct? Two DNA strands are joined to each other by no. That's present in one DNA strand. It's joined by hydrogen bonds. Okay. The alignment of bases to form a double helix is only achieved between antiparallel strands. That's true. Three hydrogen bonds are formed between all bases containing purines. No. Purines are adenine and guanine. Adenine forms two hydrogen bonds. Come on. The number of cytosine bases always equals to the number of thymine. Wrong. It should be guanine. 26. 25. A student sketched a diagram to represent the process of transcription. Which part of the diagram shows a non-transcribed strand? Non-transcribed strand. Okay. So guys, careful over here. In 25, B is the transcribed strand because look, this is what molecule exactly? This is the mRNA molecule. This can be clearly seen by the presence of uracil. So this is being transcribed. D is basically the mRNA, and C is basically a bubble, the transcription bubble, and A is the non-transcribed strand. So A is the correct answer. Which row is correct for the what's the enzyme required here? RNA polymerase. Which row is correct for the movement of water in a root? Okay, we have two pathways, the symplastic pathway and apoplastic pathway. Okay, so if you take a look at chapter 7, I guess it was 7, right? This is very important for you guys, the symplast and apoplast pathway. Apoplast means entering the cell walls. We move through the cell walls. We do not use the pores. We do not use the plasmodesmata. But in the symplast, we use the plasmodesmata. Okay, you guys need to understand something though. The apoplast pathway is blocked. Where? Due to the band of Severin, the Casparian strip in the endodermis. Okay, so apoplast pathways through the intercellular spaces. Oh, so it won't be lignin. It has to be suberin. Okay, that's why in the Casparian strip, clearly it's not lignin, right? It has to be suberin. It has to be suberin, and the apoplast pathway is closed. Only the symplast pathway is open. You guys need to understand that. Okay, only the symplast pathway is open. The apoplast pathway becomes closed. So the apoplast pathway through intercellular spaces, the symplast pathway through intercellular spaces. The maximum answer is A, but the apoplast pathway is supposed to be blocked in. Let's check this though. Wait. In a young root, the suberin forms bands in cell walls called Casparian strips. The symplast remains open. In an older root, entire cells become suberized, closing the symplast pathway too. In certain cells called passage cells, no water can enter these cells. Only the symplast remains open. This band is called the Casparian strip and goes right around the cell. It stops water through the apoplast. Okay. Simplest pathway through intercellular spaces. Simplest pathway through intercellular spaces. In Apoplast, we move from cell wall to cell wall through intercellular spaces. Oh, okay, my bad. The question just asked you. I misinterpreted the question. They just asked you like how does the apoplast pathway work? It moves through intercellular spaces. Simplest is not through intercellular spaces. And what is present in the Casparian strip? Suberin. They didn't ask you like which pathway is still open due to the Suberin strip. That wasn't the question. They just asked you like what path does the apoplast pathway take? It's intercellular spaces. Simplest takes plasmodesmata. Okay, great. So 27. The table contains some information about uptake and movement of water and of mineral ions in plants. Using the information provided, which factors will affect the uptake and movement of water or of mineral ions? So humidity of water or of mineral ions, anything works. So if it becomes more humid, there will be less evaporation. So water uptake will reduce. One is correct, and vice versa. Surface area of root hair cells will increase uptake. Oxygen concentration and temperature. So 1 and 2 are correct for sure. 1 and 2 have to be correct, so the answer is A. Basically, temperature increases evaporation, right? So more osmosis will occur. And what about oxygen? Basically, if there's more oxygen, there will be more respiration. You can think of it that way. So more CO2 will be given off. And it's like a vicious cycle. More photosynthesis will occur. So we need more water for that, basically. So what's the correct explanation for number 3? Let's see. Yeah, more oxygen concentration or decrease in oxygen concentration. Okay, let's think about decrease. If there's a decrease in O2 concentration, respiration will also decrease, right? And it does affect photosynthesis in another way, which does affect the uptake of water and mineral ions. So 3 is also correct, okay? But yeah, you could get to this answer through elimination. 28, important question. Which changes to the water potential and the volume of the solution the phloem sieve tube occur when sucrose is moved from photosynthesis and leaf into a phloem sieve tube. So this is the leaf, this is the companion cell, and this is the phloem sieve tube. So sucrose is moved from the leaf into the phloem sieve tube. So if you move sucrose, the water potential actually decreases. But as a result, water will flow in since the water potential decreased. That's why the volume increases, okay? Very common question. You'll find this every year. 29. A student wrote the following statements about a possible mechanism for loading sucrose from a source. When energy is released from ATP, the released energy is used to move sucrose through a co-transporter protein in the companion cell. Wrong. This is the companion cell, phloem sieve tube. This is the cell wall. We use energy to pump H plus inside here, but sucrose moving down through the co-transporter is a passive process, which is why A is wrong, 1 is wrong. It has to be D. As sucrose is moved into the companion cell, the pH in the cell wall of the companion cell decreases. True, because the H plus comes along with the sucrose, so the pH will Wait. One is wrong because it's a passive process. Energy is only required to pump H plus into the cell wall. As sucrose is moved into a companion cell, as the sucrose moves into the companion cell, the pH in the cell wall of the companion cell decreases. The proton pumps in the cell membrane of a companion cell moves sucrose into the phloem safety element. No, the proton pumps in the cell membrane of a companion cell move protons into the cell wall of the companion cell. Okay, so 3 is also wrong. So 2 is correct. But there's an issue here. There's a slight issue. Check this. Basically, in the loading process, what happens is we pump H plus into the cell wall. As a result, there's a high concentration of H plus in the cell wall. pH decreases, more acidic, and then passively it will come down along with sucrose. The high concentration of H plus ions drives transport of sucrose molecules. Okay, so this is co-transport. This does not require ATP. So according to our question here, as sucrose is moved into a companion cell, so as sucrose moves from the cell wall into the companion cell down the gradient of hydrogen, the pH in the cell wall of the companion cell decreases. I guess they're talking about the overall process, because this overall process does in fact cause an increase in the hydrogen ion concentration of the cell wall. But the moment the H plus comes down with sucrose, this is true that H plus does decrease from here, but this overall process increases the H plus concentration in the cell wall. So yeah, in a sense, 2 is correct. Okay, number 30. The diagram shows the transverse section through an artery. It has tunica intima, media, and externa. So intima is basically endothelium. What about media? The layer that they have marked with X. So it basically contains smooth muscles for, uh, constriction and dilatation. It has elastic fibers to withstand pressure, and it also has collagen. Okay, it has all three. This is very important. You guys need to know this. This is basically chapter 8, transport in mammals. So you need to know the structure of veins as well as arteries. All right, check this. It's just endothelium, the inner layer. Middle layer contains elastic fibers, smooth muscle, and collagen. The outer layer contains some elastic fibers and collagen. It doesn't really contain muscle. Okay, this is a very important question for you guys. 31. What is the systolic blood pressure? Interesting. So basically, the systole corresponds with the contraction of the ventricles. The systole corresponds with the contraction of the left ventricle, and it corresponds to the maximum blood pressure in the arteries. This is the one we measure in the brachial artery. Okay, when we measure blood pressure using a sphygmomanometer, the maximum blood pressure in the arteries is the systolic blood pressure, and the time corresponds with the contraction of the left ventricle. So it's not really the blood pressure of the left ventricle, but rather the pressure in the arteries, but it occurs due to the contraction of the left ventricle. Okay, 31 is C. Now we have a cardiac cycle. The diagram shows pressure changes in the left side of the heart and the aorta over time. The length of this cardiac cycle is 0.6. Points 1, 2, 3, 4 indicate when atrioventricular valves and semilunar valves either open or close. Let's see. What is the total time during one cardiac cycle that the atrioventricular valves and the semilunar valves are both closed at the same time? Interesting. So this is also present in the course book. You need a good understanding of this concept overall. Let me show you. Wait up. So you can study this picture if you guys want. While I was tutoring, I mentioned this graph. Where is it? Wait, I don't have it over here. Let me find it out for you. Here you go. This is a good picture. Yes, I'm gonna give you like 2-3 pictures to study. This is just from Google, okay? Good images. Anything else? Yeah, this works. I guess these are enough. So basically, let's study this. The blue line is the pressure of the left atrium. It always remains low. Left ventricle, the pressure in the left ventricle increases by a lot. Similar to the contraction of the left ventricle, okay? So when the left ventricle contracts, ventricle contracts, it basically opens the semilunar valves and closes the, the when the ventricles contract in general, they open the semilunar valves and close the bicuspid and tricuspid valve. Okay, the mitral valve basically in the left side, which is bicuspid, and the tricuspid valve on the right hand side. So when the left atrium actually contracts, the bicuspid or the tricuspid valve is open. See, when the atria contract over here, the bicuspid valve and tricuspid valve are open. Okay, that's the contraction of the left atrium. Now afterwards, when the uh, ventricle starts contracting, there will be a moment when basically the lub dub, these sounds, the lub is actually due to the closing of the atrioventricular valves or the tricuspid and bicuspid valve. So there will be a moment where, look here, just when the ventricle starts contracting, the atrioventricular valve will be closed and the semilunar or the aortic valve will also be closed during this time span. But afterwards, the semilunar valve will open and blood will flow through the aorta. Okay, now the semilunar valve is open, but the atrioventricular valves are closed. Afterwards, when the pressure in the ventricle drops, there will be a time when, due to the decrease in pressure, the atrioventricular valves will close again, and the semilunar valves will close again, and the atrioventricular valves are still open. But when the atria starts contracting again, when the atria start filling again, what happens basically is that due to the increased pressure in the atria, the atrioventricular valves open again, but the semilunar valves still remain closed. So essentially, what happened here? The atrioventricular valve closed, but the semilunar valve was still closed. At this moment, the atrioventricular valve was closed, but the semilunar valve opens. So in this time span, both were closed. Here, the semilunar valve closes, but the atrioventricular valve is still closed. So both of them are closed, but in this moment, the atrioventricular valve opens due to atrial filling, increased pressure in atria. The ventricles will fill, but the semilunar valve is still closed. So I'm mainly looking at these two time spans where both the valves are closed. Okay, hopefully I could clarify. See if this makes sense. I tried my best. So mainly I'm looking here basically from this time span to this time span over here. In this time span, both are actually closed. Similarly here, it's closed, it's also closed here. So basically this part and this part. So this is 0.12 to 0.12345, 0.15. That's 0.03 seconds on this side. and here it's 0 0.3, 1, 2, 3, 4, 5, 6, 0 0.36 to 0 0.4, which is 0 0.04. So 0.04 and 0.03, that is 0.07. We should go with C. Okay, 32 is C. Great. Which reactions take place in the capillaries surrounding an alveolus? Okay. Carbon dioxide plus water equals to carbonic acid. Impossible. Because you need carbonic anhydrous to catalyze this reaction. That is only present in RBC. One is wrong. Carbon dioxide plus hemoglobin. Carbamino. Oh wait, do they do they want us to look for RBC? I'm actually a bit confused. Let's see though. It's basically happening in an alveolus. So this is the alveolus. This is the capillary. We have the RBC over here. Typically, what happens in the alveolus? O2 comes from here to here, and CO2 goes from the blood to the alveolus. So, oh, they want us to think of it in this way. I get what they mean. Basically, guys, since here we want carbon dioxide to exit, we are in fact going to break down H2CO3 to form water and carbon dioxide, okay? And oxygen will come into the RBC, bind with hemoglobin to form oxyhemoglobin, HbO8. Also, one other thing will happen. Not carboxyhemoglobin, carb amino hemoglobin will break down. Carb amino hemoglobin, carb amino hemoglobin will break down to form CO2 plus Hb. Okay, these are the reactions that's occurring in an alveolus, and in a muscle the opposite will occur. It will be from right to left rather than left to right. Okay, so carbon dioxide plus water to carbonic acid, wrong. It's the other way around. This happens. Carbon dioxide plus hemoglobin to carbon and hemoglobin, wrong. It will be the other way around. So 1 and 2 are actually wrong. Okay, hemoglobinic acid. Okay, let's talk about these two, 3 and 4. We need to figure out which ones are correct. So hemoglobinic acid. Where does this usually form? This usually forms in the muscles where there is lactic acid production, and it actually leads to the Bohr effect, the Bohr shift, which shifts the curve to the right hand side. So which allows more oxygen to be lost. But the reverse Bohr shift, or this is called the hamburger. This is the hamburger shift. The hamburger shift occurs in the alveolus. So basically in muscles, what happens? Basically hemoglobin combines with acid to form hemoglobinic acid, which allows more O2 to be released. In alveolus, the opposite occurs. Hemoglobinic acid will break down to form hemoglobin and H plus, which shifts the oxygen saturation, the PO2, partial pressure of oxygen, the hemoglobin oxygen saturation curve to the left hand side. So 3 is in fact correct. And number 4, hydrogen carbonate ions plus hydrogen. Oh yeah, H plus plus HCO3 minus does form H2CO3, which again forms H2O plus CO2. This is correct. 3 and 4 are in fact correct. 33 is B. Good question. Last ones. Which statement explains the importance of the chloride shift in RBC? Okay, basically it maintains ion balance. You know that in the RBC, a lot of HCO3 minus is produced. So that gets out of the cell, right? Most of the carbon dioxide is transported in blood as HCO3 minus bicarbonate. But there will be a deficit of negative charge inside the cell, right? That's why chloride ions enter the cell to maintain balance. That's basically it, honestly. So hydrogen carbonate ions diffuse into plasma from RBC, and chloride ions diffuse into RBC to maintain a balance of positive and negative ions. Okay. Next, the photomicrographs show a cross section through the lining of the part of our respiratory system. Great. The respiratory epithelium is pseudostratified ciliated columnar epithelium. Okay. So let's look at 35. Goblet cells are visible between squamous epithelial cells. Smooth muscle is visible. The section cannot be a bronchial as cartilage is visible. Honestly, this is hyaline cartilage. This is probably it's a part of the trachea. It looks like the C-shaped cartilage. Okay, and clearly the smooth muscle is visible, guys. Look, I'm going to show you a photomicrograph. Wait, photomicrograph of trachea. Check this. with labeling, then it'll make sense to you guys. Wait up. Can't find a good enough picture, honestly. Is this good enough? Wait. See if you guys understand these. This is a much more, this is a picture with greater magnification. Mm, I'm trying to find a better picture for you guys. So yeah, honestly, basically the layer, this is the ciliated columnar epithelium over here. We zoomed in over here. This is the cartilage, basically. This part is the cartilage. I can understand that by looking at the lacuna. See the lacuna? So these are the lacuna zoomed in. This is hyaline cartilage, basically. So we have the mucosa, which contains the lining epithelium, connective tissue. We have the submucosa, which has some glands just below. So it can be a bronchial because only cartilage is only present in the trachea and bronchus. Okay, trachea and bronchus. So basically 3 is correct. This is a correct fact. I'm gonna go for 3. Smooth muscle is visible here. If you zoom in on this figure, this is the adventitia, the last layer. This is the adventitia, but you can see that there's smooth muscle over here. Can you see that? There are glands over here. This is the lining. There are glands. There's smooth muscle and vessels over here, as you can see. Smooth muscles always lie below the lining epithelium. Okay, like if in mucosa there's a layer called lamina propria and muscularis mucosa. So there is smooth muscle over here if you zoom in. This is not the muscle layer, by the way. This is the adventitia. It's like a covering. So the smooth muscle is over here, and these are the vessels. Okay. So 2 is also correct. Goblet cells are visible between the squamous epithelium cells. So these are just cirrostratified ciliated columnar epithelium. What does a goblet cell look like if you're curious? Let me show you a picture. Goblet cell photomicrograph. Does the course book have a better picture? Right. Chapter 9, right? So yeah, you will only find cartilage in these 2 parts and goblet cells as well. But how to identify a goblet cell? Goblet cell. Yeah, it's basically a goblet shape, but wait. So basically, We can see this in our photomicrograph. The goblet cell won't have cilia, and basically it will have droplets of mucin. There will be a lot of staining. Could I clarify? And it's actually goblet shaped, so it will be more thick on the upper end, on the upper portion. Could I clarify? Here, no one's really thick on the upper portion. Everyone has cilia, and the nucleus is located over here. And no one's really thick on. It's not. No cell looks like this. No cell looks like this. There's no goblet cell here. Okay, so we cannot see them between. It's not visible. That's why one is wrong. We're gonna go with 2 and 3 only. I couldn't really find a picture for goblet cell online. Wait, let me see if I can find one. Give me a bit. uh Goblet cell in trachea. Basically this is what it looks like. Come on, see this. The mucin is clearly visible here. It would have looked like this, but we don't see anything on the top, right? On the staining, on the on the photomicrograph. Okay, so that's fine. It's clearly, you can clearly identify it. All right, anyway. 36. The surface tension of the layer of liquid lining the alveoli tends to pull the walls inwards, so alveoli could collapse. That's why we produce the fluid surfactant, which prevents the collapse. Okay, so mainly it's surfactant. So this chemical is called surfactant. This is the main factor, and it's the only factor, honestly. So surfactant prevents it. It is secreted by alveolar macrophages. Okay. So when children are just born, after 28 weeks they can produce that, so it's a viable birth. 37. What will reduce the rate at which bacteria become resistant to antibiotics? Prescribing two antibiotics with different modes of action. That is true, so the bacteria, we can be more confirmed that the bacteria dies. Finishing a prescribed course. True. Prescribing different antibiotics for the same bacterium. True. If you keep on prescribing the same antibiotic, the bacteria will get more exposure to that and might develop resistance. All right, last three. T lymphocytes have a protein PD-1 on their surface. Some cancer cells have a receptor molecule on their surface which binds with PD-1, inactivating the T lymphocyte. Okay, so for 38, T lymphocytes have a protein PD-1 on their surface. Some cancer cells have a receptor molecule on their surface which binds with PD-1, inactivating the T lymphocyte. A monoclonal antibody is lambrolizumab. Lambrolizumab or iambrolizumab. Looks like it's an I basically. Iambrolizumab has been produced against this receptor. So this is the lymphocyte. It has this receptor called PD-1. So we have a drug called iambrolizumab which actually binds to this. Trials show that 54 of 135 people with advanced skin cancer who were given iambrolizumab, the tumors more than halved in volume. So this is the lymphocyte. The protein is PD-1. So the cancer cell basically has a receptor on their surface. This is the cancer cell's receptor, which actually binds to our uh, PD-1 on lymphocytes. As a result, the lymphocyte becomes inactivated. But if we administer the drug, the monoclonal antibody, iambrolizumab, it actually binds to the site PD-1, so the cancer cell cannot bind. As a result, the inactivation does not occur, and the people who are given ambrolizumab showed that, like in 54 out of 135 people, the tumors more than halved in volume. In 6 of the 57 people who were given the highest dose, the tumors even disappeared. Okay, what may be correctly concluded from this information? Oh wait, it was Lambro. This was a small little my bad. So lambrolizumab binds with a receptor on the surface of skin cancer cells. Oh wait. I misinterpreted the question. The monoclonal antibody is produced against this receptor. So not against PD-1, but the receptor of the cancer cell. Okay. So my bad. So this is the receptor on the lymphocyte. Sorry, the protein PD-1, and this is the receptor on the cancer cell. So the monoclonal antibody lambrolizumab, basically this is the drug which binds to the receptor on the cancer cell. As a result, this can no longer bind to the bind to PD-1 on lymphocyte, and the cancer cannot inactivate the lymphocyte. So that's how it works. Okay, so lambrolizumab binds with the receptor on the surface of the cancer cells. Uh, this is correct. This is fine. Cancer cells to which lembrolizumab is bound cannot inactivate T lymphocytes. That is also true because since it's bound to this receptor, it can't bind to PD-1. 2 is true. Lembrolizumab targets and kills cancer cells. So we aren't actually sure about the mechanism of 3. We just know that it binds to the receptor so that it cannot inactivate lymphocytes. Lembrolizumab allows the patient's own immune system to kill cancer cells. This is true. Okay. Now the lymphocyte can be activated, so which can kill the cancer cell. So 38 B is the correct answer. Good question. 39. A person's blood group is determined by the presence of antigens. On the RBC, the table shows the antigens and antibodies. During a blood transfusion, it is essential that the person receiving the blood does not have antibodies to the donor's blood. Which blood groups can be given to a person with blood group AB? So you guys got to understand, for a person with blood group AB, you have both A and B antigens, so you have neither antibodies. That's why the people with AB blood groups are superior. So they can take blood from any donor because they don't have any antibodies. The recipient does not have any antibodies. Okay, so we don't actually look at the donor's antibodies, but rather we look at the recipient's antibodies. Since the recipient does not have any antibodies, so it doesn't matter which blood you give, all of them should work. But in recent times, we don't follow this method. A person with AB blood group is usually given AB blood group, but it's fine if you give the other ones as well. Okay. Which types of cellular stim? So in O blood group, we have both antibodies. That's why a person with O blood group can only receive O. And for someone with O negative blood, it's really harmful. It's really risky because it's very rare to get O negative, right? Which types of cells are stimulated to divide by the cytokines produced by T helper cells? Okay, so T helper cells, Th cells, there are Th1, Th2, Th17. Basically, in med school, I study immunology. It's much more detailed there. But A level immunity has helped me a lot. Like it helped me through the process. I knew the basics beforehand. So according to our syllabus, T helper cells stimulates B lymphocytes and T killer cells. Okay, the helper cells stimulate T killer cells, cytotoxic T cells, and B lymphocytes. They cause their proliferation, right? But T helper cells also do stimulate macrophages in a way that's more complex. You don't need that. So according to your syllabus, just know that T helper cells stimulate B lymphocytes and T killer cells. Okay, so that is all, guys. Hope you liked the video. If I did make any mistakes, please feel free to write that down in the comments. I'll fix that then. Remember to subscribe to the channel if you like the content. I'll link the playlist for bio paper 1 up here, and when I solve May June 2022 paper 1 2, I'll link that up here, and when I solve May June 2022 1 3, I'll link that down here. Okay, see you guys.